Hello friends, I am Dr. Firuza Parekh. I am the director of the Fertility Jaslok International Fertility Center. Today we are going to talk about genetic testing of embryos by a process called Preimplantation genetic testing, PGT for short. Every year an estimated 7.9 million infants, that is about 6% of worldwide births, are born with serious birth defects. Although some congenital defects can be controlled and treated, an estimated 3.2 million of these children can be disabled for life. A genetic disorder is a disease that is caused by an abnormality in an individual's DNA. Abnormalities can be as small as a single mutation or change in just one gene, or they can involve the addition or subtraction of entire chromosomes. Most genes will come in pairs, one from the mother, that is from the egg, and one from the father, that is from the sperm. When the function of a gene is altered by a change called a mutation in a specific sequence, a genetic disease results. These mutations can be transmitted in families from generation to generation, that means they are inherited, or there can be a new change in an individual, and that is called a de novo change. These mutations lead to either single gene or multiple gene disorders. PGT, that is preimplantation genetic testing, is the method used to identify embryos for the presence of any known genetic defect prior to implantation. This testing thus prevents the passing of genetic abnormalities from the parents to the newborn through assisted reproductive technology. During the IVF cycle, a single cell or few cells are removed, that is, they are biopsied from the embryo, and they are checked for certain genetic abnormalities. The normally reported embryos are then transferred back to the mother's uterus. So let's look at what chromosomes are. Every cell of the human body has 46 chromosomes, which are a basic genetic material. The egg and the sperm contain half the number of chromosomes, that is 23 chromosomes. After fertilization of the egg with the sperm, these chromosomes unite and form the first cell of the embryo with 46 chromosomes. If the embryo has all cells with 46 chromosomes, it is termed as euploid. If there are more or less than 46 chromosomes in the embryo, it is termed as aneuploid. The loss of a chromosome is called monosomy, and the gain of a chromosome is known as trisomy. Thus, to check the gain or loss of chromosomes fully or partially prior to implantation, we can use preimplantation genetic screening, which is now also termed as PGT-A. For PGT, the couple has to undergo IVF so that the embryos are prepared and cultured outside the body before implantation. Generally, on day 5 of the embryo development, that is when it is at the blastocyst stage, 4 to 5 trophectoderm cells are taken from the embryo. This is done by a very delicate process of embryo biopsy. These cells actually will form the placental cells, and therefore taking these cells from the embryo will not damage the embryo. Once the results of these testing is available, the genetically normal embryo can be transferred into the mother's uterus. Thus, the aim of PGT is to have a genetically normal baby. PGT helps to improve pregnancy rates, decreases the incidence of miscarriages, and is very helpful in older age women and in couples who have had previous failed IVF attempts, history of spontaneous miscarriages, and couples who are carriers of known genetic disorders. There are different ways of testing for this genetic testing, and some of the techniques are array CGH, next generation sequencing, FISH, PCR, and karyomapping. A proper technique should be chosen depending on the genetic abnormality that is to be tested. We have helped couples with several genetic problems to conceive. PGT is used in cases of thalassemia and sickle cell anemia, then neurological diseases such as Huntington's chorea, Duchenne's muscular dystrophy, hemophilia, neurofibromatosis, Leigh disease, and many more. At Fertility, it is our mission to help parents have genetically normal and healthy children.